Okay. Thank you very much for inviting me. I hope everybody can see everything now. And yes. Sorry for the hiccup. It's the first time that I use GoToWebinar, so yeah, there was a couple of technical hiccups. Anyway, so today I'm gonna talk about major QTLs that we found for resistance against a bacterium called Flavobacterium psychrophilum in Atlantic salmon. And I'm a bioinformatician. I work in a company called Aquagen, which is an international company, but the research and development, or part of the research and development, is located in Norway, and that's where I'm sitting currently. And this is, of course, not only my work, but it's kind of one part of Aquagen's big research and development work. And of course, there are many other research institutions and people involved, which I will recognize later. So just to give you a little bit brief background. So just a second. There's something not on here. So like this. So the disease that I'm talking about is caused by a bacterium called Flavobacterium psychrophilum. This is the cause for flavobacteriosis in coho salmon, Atlantic salmon, and rainbow trout. The bacterium is most prominently known from rainbow trout, where it causes a disease called rainbow trout fry syndrome, or bacterial cold water disease. These diseases are one of the main causes for mortalities and economic loss in rainbow trout. However, this disease has recently also been isolated from Atlantic salmon fry, so fish smaller than approximately 1 gram, in Scotland following disease outbreaks. And this is caused and concern for the industry and led to the initiation of this research project. Important is here also that the bacterium usually affects fry and larger fish in freshwater hatcheries and on-growing sites, and the fish are normally too small to be vaccinated or to have received the vaccination. That's one point. And the other point is that, to my knowledge, there is no commercial vaccine available. And for that reason, antibiotics are often used to treat the affected stock, which is, of course, not kind of perfect from an environmental perspective. So since Aquagen, since we are a breeding company, we make a living by selling eggs to producers. And so our idea was to use, to try to find out if we can use genetics, if we can use selective breeding as a remedy for this disease in Atlantic salmon. So to test this, we conducted 2 challenge trials using fish from the Aquagen nucleus. And for both trials, we used a newly developed challenge model from the University of Stirling. So I'm not going to go too much into details, but basically it's a bacteria that enters through the skin. And so the challenge trial kind of, or the challenge method is kind of like contains the stressor part and then immerses the fish into the bacterial and then kind of mimics the natural route of infection, which is through the skin. Also very important for this experiment was that in both trials we used different isolates of the bacterium, of Flavobacterium psychrophilum, and those isolates originated from natural outbreaks in Scotland. The setup was a little bit different. So we used, the first trial was conducted in 2018 with approximately 3,000 fish in 2 parallel tanks, so 1,500 fish per tank. And in 2019 with approximately 1,200 fish in 1 single tank. The cumulative mortality you can see here. So the mortality starts approximately 3-4 days post-infection and then kind of peaks out approximately 2 weeks plus minus after infection. You can see that the cumulative mortality was higher in the trial we're using the 2018 population. And you can see that there was a slight difference between the 2 tanks that were used, indicated here by the 2 red lines. But on average, it was pretty close and had a cumulative mortality of 27%. 2019 was a little bit lower, around 19%. So then we were, of course, interested if there was a genetic heritable component to this disease. So we genotyped all the fish. So this is, of course, how we do it. During the challenge trial, we collect the mortalities twice a day, isolate DNA from tissue samples, and at the end of the experiment, we call all survivors and genotype them as well. So that means in total we have genotyped over 3,000 animals from the 2018 trial and over approximately 1,200 animals from the 2019 trial. All these fish were then genotyped on the Atlantic salmon 70K thermofisher axiom array, containing actually 72,000 SNPs distributed approximately equally across the Atlantic salmon genome. Then we did some quality filtering, etc., missingness, etc., and ended up with approximately 56,000 markers that we could use to estimate genetic heritability. Genetic heritability was significant and quite decent, actually, with 0.46 for the 2018 trial and 0.37 for the 2019 trial, where the overall mortality was slightly lower. However, we were, of course, interested if the genetic component that we're measuring in both trials was the same. So we tested for genetic correlation between the 2018 and 2019 trial and found that there was a high genetic correlation of 0.94, which was very nice and reassuring. So then, of course, as you do, we conducted the GWAS, a genome-wide association study that my colleagues talked about before me. Unfortunately, I didn't understand so much since I don't speak any Spanish. But this is, of course, a GWAS. So on top here, the top plot shows you the GWAS results from the 2018 trial and the bottom results from the 2019 trial. The physical positions of the SNPs along the Atlantic salmon chromosome are shown on the x-axis, and the significance of the SNPs in an inverted scale on -log p is on the y-axis. What you can see here is, of course, quite dominantly that we have a very strong and very significant peak, nice and small, on chromosome 9. And we also have a quite significant and nice peak actually here on chromosome 5. It is a little bit dwarfed since the one on chromosome 9 is kind of like that huge in the 2018 trial, but it's nevertheless very significant and has also kind of like a nice shape. I should probably also mention, so the dotted line represents the cutoff for the Benjamini-Hochburg correction, and that is the dashed line is the FDR correction. Then on the bottom, you can see that kind of the same peak appears on chromosome 9 and on chromosome 5, whereas the peak on chromosome 9 loses a little bit of its significance. Sorry, that was the dotted line is of course the Bonferroni correction of that. Anyway. So then, just to show you kind of that the peaks are indeed actually the same SNPs. So this is just a scatter plot showing the significances, the -log p values from the 2019 trial on the y-axis, and the -log 10 p values from the 2018 trial on the x-axis. And what you can see is that the top SNP on chromosome 5 and the top SNP on chromosome 9 are indeed the same. So this is a good result. Shows us that the top SNPs are reproducible and especially also using different isolates of the bacteria. So that means that we kind of have identified, or we have identified actually 2 potential QTLs. So now the next question that we are trying to answer is if there is any benefit by combining the information from these QTLs for selection purposes. And this is what I'm trying to show here in this plot. So there are 2 panels. There's the results from the 2018 trial on the left and from the 2019 trial on the right. And then we have basically a bubble plot. So each bubble is a combination of the alleles for the QTLs on chromosome 9 on the y-axis and chromosome 5 on the x-axis. The size of the bubbles is proportional to the number of fish with this combination of alleles. Whereas the color scale, kind of like if it's pink or blue, indicates the proportion of mortalities or survivors with this particular combination of genotypes. And what you can see basically is that if you are a fish that ended up with all the bad alleles from both QTLs, the one on chromosome 5 and the one on chromosome 9, indicated by the small q sign, so small q here and small q here, then your chance of surviving a Flavo infection is actually pretty low. Whereas if you're a fish on the other spectrum that received both good alleles for QTL on chromosome 5 as well as the QTL on chromosome 9, then your chances of surviving a Flavobacterium infection are actually significantly higher. So to conclude this really quickly is we have identified two major QTLs for resistance against Flavobacterium psychrophilum in Atlantic salmon. Both QTLs have been confirmed in two independent challenge trials, with 2 different bacterial isolates. And as a little bit of commercial, I can say that Flavo-resistant eggs are actually now available from Aquagen. However, there's of course, as it is, there's always a lot of things left to do. We are currently working on a field validation for those markers. Furthermore, we are analyzing RNA-seq data from approximately 60 animals that were taken from a parallel challenge trial, from a time series challenge trial. And we, of course, hope to fine map and eventually using the RNA-seq data to hopefully identify the causative gene / causative mutation. But that we'll find out soon. And of course, finishing the manuscript. Then, last but not least, I would like to thank everyone that was involved in this project, which is first the Scottish Aquaculture Innovation Centre, which financed this project. Then Cook Aquaculture, which is a partner in this project. And of course, the main academic partners or the research partners, which is Aquagen and University of Stirling, which have helped to make this project possible. And of course, I would also like to thank the organizers of this event for inviting me to this venue. Thank you very much.